Hello and welcome back to Educator.com. Today we're going to talk about the topic of transcription. As an overview, we're going to talk about eukaryotic transcription. Then we will also talk about how that differs from prokaryotic transcription. And then we will focus on some of the post-transcriptional modifications that are seen. So the RNA processing, splicing, and editing. So, what is eukaryotic transcription, or what is transcription in general? That is the making of RNA from a DNA sequence. So for eukaryotes, we're going to talk first, and we have RNA polymerase synthesizing RNA using one strand of DNA as a template. So this is the first step of gene expression, and transcription is responsible for making rRNA using RNA polymerase 1, mRNA using RNA polymerase 2, and tRNA using RNA polymerase 3. This is a three-step process, and it involves initiation, elongation, and termination. We'll talk about each one of these in depth, but let's first kind of show you what a transcription bubble would look like. So here's our DNA. This would be what we would call our, let's write in our RNA first. In this case, I'm saying mRNA. Okay. Okay. Okay, so what we have here is the mRNA, okay, and we will be making the proper hydrogen bonding. All right, so we have DNA, we have it opened up, and this is what is called our replication bubble. And this is about, it can vary in length. So what we have our DNA opened up, and we have our mRNA being synthesized off of a strand of the DNA that is acting as the template strand. So this right here would be called our template strand, and it could also be called it has antisense. Okay, and this is what's called our coding strand, and this has sense. And sense is also known as positive, antisense is known as negative, okay, orientation. Now our mRNA will be sense as well, okay, because it's going to be complementary to the template, so it'll have to be the opposite. Okay, so sense or negative. All right, and by the way, this is all occurring inside of our RNA polymerase, right? So big RNA polymerase going through, right? And since this is mRNA, this is RNA pol 2. Okay, so this mRNA, as I said, will be a sense or negative strand. Which is why we can talk a little later on in these units, why we can use antisense RNA or siRNA, silent interfering or small interfering RNA, which can bind to and degrade mRNA, which can be a way that we can regulate gene expression. Okay. Now, let's continue on. So, let's talk about eukaryotic transcription. We have the transcription start site, which we call the TSS, is being found at the +1 position. Okay. The promoter in most of our regulatory regions are upstream or to the negative side, and our transcription and termination regions are downstream or to the positive side. Okay. And our transcription unit, the entire unit, extends from, the promoter to the termination region. Okay, and it will include such things as a GC box, a CAT box, TATA box, BRE initiator, DP. And I'll tell you what each one of these are in the following slides.